morning and thank you everybody. Today I'm going to present one part of my PhD work, the characterization of the putative polysaccharide gene cluster and multidrug-resistant Enterococcus faecium to develop a targeted therapy or vaccine. Actually, the Enterococci or Enterococci is basically gram-positive bacteria, and these are anaerobic and produce lactic acids. And in past, both E. faecalis and E. faecium were considered a low-grade pathogen, but nowadays they are going to be more resistant, and these are nowadays the emerging multi-drug resistance. And this caused urinary tract infection, bacteremia, endocarditis, especially in the immunocompromised or hospitalized patients. Recently, the Center for Controlled Diseases reported that 38% of the hospital-associated enterococcal isolates identified on the species level were Enterococcus faecium. So Enterococcus faecium is nowadays more prevalent as compared to Enterococcus faecalis. So if we look the cell envelope, cell envelope of the Enterococcus faecium, it is quite interesting but too complex, and still the cell wall of the both E. faecalis and E. faecium is still ill-defined. Here we see the lipoteichoic acids and other extracellular surface protein such as wall teichoic acid. All these surface protein play important role to the disease pathogenesis directly or indirectly. So interaction of E. faecium with the intestinal epithelial cell. So actually what happened, patients in the hospital are administered broad spectrums of antibiotics which increases the hospital-associated E. faecium colonization of the GIT tract. Actually, the antibiotics down-regulates the expression of REC3 gamma, and this is basically a toll-like receptor which provoke an attack on the target, the peptidoglycan of the most of the gram-positive bacteria, and so and reducing the infection. But in this case, in immunocompromised or hospitalized patients, these receptors is down-regulated, and because of the lipopolysaccharides, which is down-regulated by some antibiotics indirectly, not directly, and this happen, and this increase the outgrowth and intestinal colonization and spread of E. faecium in the hospital infection and hospital environment. So capsular polysaccharide is a vaccine candidate for enterococcal infection. As I already told, the capsular polysaccharides are still ill-defined and the mechanism is still not known, particularly in Enterococcus faecium. So the conjugated vaccines nowadays available for the Streptococcus pneumonia, influenza, etc. The aim of my project or our project is and was to develop an antibody against this enterococcal faecium infection. So to screen the loci for the capsule in the genome of this strain, this is basically the clinical strain, and it is more prevalent, especially in the urinary tract infections, so U0317. So we screened more than 300 contacts, 342 contacts, and we found two interesting contacts. One is 22 kilobase pair and another one is 19 kilobase pair. The entire locus was 41 kilobase pair, and it contains more than 35 genes, and including 11 glycoside transferases. But we focus on the downstream part because the 22 kilobase pair part, it shows strong homology with the other known sequence of Enterococcus faecium. So we focus the downstream part, which is 19 kilobase pair, it contains 17 genes, including the capsular polysaccharide biosynthesis protein, which is annotated as a CAPD, and it shows homology with the Streptococcus pneumonia CAPD gene. And this protein contains 336 amino acids on 1,000 nucleotides, and function is N-linked glycosylation. So we focus our study on this polysaccharides protein. So we create a knockout mutant. Here we amplified the downstream gene and upstream gene, and we deleted the 1 kb region of the CAPD from the middle, and we re-amplified these 2 kb products and used PLT06 vector as a shuttle vector, which is 7.9 kilobase pair, kilobytes. And we use here spectinomycin as a acid as a selection marker. And after electroporation and homologous recombination in the genome of the Enterococcus faecium U0317, we confirm the deletion of CAPD by PCR sequencing and as well as by Southern blotting. So here are some PCR. Here is our 1 kb, the downstream and upstream gene, and here is our answer containing the spectinomycin acid. It is 3 kilobase pair, and here is our 2 kb construct, the deleted CAPD genes, and the shuttle vector PLT06, and this is our complementation. And for the complementation, we use the double crossing over. And here the faecium, it is our control. So we also investigated the structure of the CAPD. We use the electron micrograph, and this shows the phenotypic analysis. And both our wild type and mutant contain polysaccharide and a normal cell wall structure architecture, but here in CAPD mutant, we see it is little bit more rounder and wider as compared to the wild type. So we also investigated the effect of the CAPD gene on the growth kinetics. And we saw the reduction in the CAPD genes, and it didn't reach the stationary phase. And in all 4 media, all 4 growth medium, we investigated more than 2 folds in both in chemically defined media and the natural urine. And we also seen a reduction of the CAPD genes and the TSB, which is 1.24 to 3-fold and more than 1.5-fold reduction in the BHI medium, growth medium. So our result shows that the deletion of the CAPD genes reduce the growth in all culture media. We also investigated the role of the CAPD in the biofilm production and the hydrophobicity, and it was little bit quite interesting and contradictory results, as biofilm and hydrophobicity and the microbial infection are directly proportional to each other. But here in this case, we saw higher hydrophobicity in our CAPD mutant, which is round about more than 25%, and we saw our 2-fold reduction or even less than 50% reduction in the biofilm production. But we already investigated in our previous work that the E-faeculus produce more biofilm as compared to E-faecium. E-faecium is famous for poor production of the biofilm. And also recently, work published in Australia, they also investigated that the concentration of the glucose is also important. If there is a less concentration of the glucose, the biofilm production, especially in Enterococcus faecium, are also less. So antibiotics resistance. So the cell wall of the E. faecium is an important target, as we know, and the susceptibility result of our KEPD mutant compared to the wild type and the complemented strain were similar, except for the linozyte, for which the KEPD mutant shows more resistant. We also purify the cell wall associated polysaccharides from the both complemented strain, KEPD mutant, as well as for the wild type. And we digested this cell wall associated extract with lysozyme and mutalinosine to depolymerize the peptidoglycan. And so here our result shows the wild type and the complemented strand revealed a broad band at 60 to 100 kilodalton as compared to our mutant wild type. It didn't migrate it on the SDS page during past staining, and it was migrated distinctly more slowly. And we also confirm our previous hypothesis that this is because of the net loss of a negatively charged motif because of loss of 2-teichoic acid. So, and we also confirm it by Sykes exclusion chromatography. The results are not here, that it is not because of the size, it is because of the net loss of the charge motif. First, it was hypothesis that maybe it is because of the charge as well as because of the size. But it is confirmed it's not because of the size, as we already deleted a mutant, 1 kb region. So we also did some opsonophagocytic assay, and we also confirmed another hypothesis, which was that this clinical strain U0317 is not sensible to the lipoteichoic acid, the anti-lipoteichoic acid. So we raised rabbit anti-sera against the whole bacterium, and as well as for the KEPD complements and KEPD mutant in different dilution, 1:100 and 1:200, and we saw more than 80 up to 90% killing in the KEPD mutant as compared to the complemented mutant and the wild type. So this shows that in the complemented mutant and the wild type the capsular polysaccharides components acts as a mass component to most of the gram-positive bacteria. We also use the cell culture model T24 cells from the human carcinoma and COCO2 cells from the colon carcinoma of the mice, and we saw more than 50% less reduction in the KEPD mutant in both cell lines. So the KEPD didn't adhere to the cells. It may be because of the bladder, as we already published that the E. faecalis didn't persist in the bladder due to the mechanical force in the flow of the urine. We also use the in vivo. We also did in vivo analysis and check also the virulence of the KEPD mutants. So we infected the 6 to 8 weeks albino mice, 10⁻⁷ CFU through vein, and we see the KEPD expression affect the bacteria survival in the bloodstream and higher number of the colonization in the liver, but we didn't saw any difference in the kidney. So in the red endocarditis and urinary tract infections, we didn't see any significant change. So this is because urinary tract infection, the epithelial cells are different and they have different adherence power, and as compared to sepsis model. So after sacrificing of the mice after 24, 48, we also did 72 and 96 hours, but we didn't saw any change. We also predict the hypothetical structure of our CAPD mutants, and this secondary structure shows 8 helices and 8 strands, and it's belong to the family NAD binding Rossmann domains and tyrosine-dependent oxidative-reductase family. But here, the helix 8 and helix 4, these are highly exposed. The other strains are buried in nature, but these are highly exposed because of the osmotic pressure or some extracellular interaction. So conclusion. So in this study, we identified polysaccharide biosynthesis protein and UO317, and disruption of these genes led to reduced growth, phenotypic alteration and structure, increased hydrophobicity, reduced biofilm formation, and increased susceptibility to the opsonophagocytic killing. So role of the CAPD CAPD mutant as a virulence demonstrated that decreased binding to the uroepithelial cells, both colon carcinoma and human bladder cells, and also lower virulence in the mouse sepsis model, especially in the kidney, but not in the blood. And UTI model, urinary tract infection, and rat endocarditis. So we saw high bacteremia during the blood infection, and as well as high number of the colonization in the liver also. So we need further study are needed to elucidate the function of the capsular polysaccharide synthesis gene clusters and its involvement to the disease pathogenesis to develop a targeted therapy to prevent this multidrug-resistant faecium infection. So at the end, I will thanks to my supervisor, Professor Bloom, and Dr. Turkan, and the funding agencies of DAD and German Ministry of Science and Technology. Thank you so much.